I want to welcome everyone to our latest Cyber Series lecture. Just as a reminder, starting with the COVID pandemic last spring, we've begun to invite some of our own Mount Sinai neuroscientists to speak to our community. It's turned out to be a great event, I think, because we're now learning more of what our own faculty are up to, which has really received a great deal of accolades. So today it's a great pleasure to introduce Dr. Rita Goldstein, who's been a close colleague of mine ever since she joined Mount Sinai. I believe, Rita, since like 2013 or so.
Exactly, yeah.
And Rita is a national leader in using brain imaging and other approaches to understand the neurobiology of cocaine and other addictions in humans. So Rita, thank you so much for joining us, and take it away.
Thank you very much for the invitation, Eric. And let me share my screen and let me know if you can see it.
Yes, that looks good.
Okay, great. And I would like to tell everybody that you can ask questions anytime, okay? There's gonna be natural type of pauses in the talk, but also anytime it's fine with me. I'm a little nervous actually, more than when I go out there to give a talk to my own colleagues and friends. And I was trying to make a focus on translational aspects. So I'll mention animal studies quite a lot if I can. I do want to also acknowledge the stressful times we live in, and I hope that science and logic and humanity and democracy will prevail. Okay, and I want to introduce our NAR group, the Neuropsychoimaging of Addiction and Related Conditions. This is some of us, including some of our postdocs and clinical research coordinators and the lab manager. Some people are not in the picture. We are socially distancing, so didn't have a time yet to take a picture. I will talk about some of our most current projects, and they're listed here. Do you see my mouse? Or not?
Yes.
Yeah, okay, good.
Mouse, fine.
Okay, good. And I would also like to mention that Nelly and I have been leading this group for many years now, a second decade, and mentoring is extremely important for us. And we have several trainees in the lab who had their own individual fellowships, including F32s and K awards, all the way to R grants. And some of them are now leading their own labs, including in Mount Sinai, Muhammad Parvaz and Karen Bachi, and also people who are leading their labs in other institutes, Stony Brook University, University of Minnesota, and Rutgers University. Okay, so here, this crowd knows a lot about addiction, or many people know a lot about addiction, but I would like to remind people that addiction is not bad habits. It's not just drug abuse or even drug dependence. So all of these three are part of addiction. They're part of the phenomenology of addiction, but only in addiction you have this severe loss of control over drug intake and drug use frequency and amount, despite catastrophic consequences to the individual's life, and despite a decrease in pleasure that's derived from the drug. In the IRISA model of drug addiction, we emphasize that in order to understand the clinical symptomatology of addiction that has to do with drug reinforcement, craving, binging, and withdrawal, we have to study impairments in response inhibition and salience attribution, where the salience, the value of a drug and drug-related cues, overpower the value and salience of everything that's non-drug related, with a concomitant decrease in the ability to control and regulate behavior. And we emphasize the importance of the prefrontal cortex in these higher order executive functions in understanding addiction. So expanding the common view, mostly from animal studies, of addiction that looked at subcortical regions and subcortical reward and habit type of processing to these higher order executive functions and the role of the prefrontal cortex in drug addicted individuals. And this focus has been very impactful, and this model summarized now in 3 different places. Okay, but let's see what's happening now in the field. And of course, the COVID-19 is something we all are extremely concerned about. And now that we defined what it is that substance use disorder. And this is a study that was led by Nora Volko, whom I did both Nelly and I, we did our postdoc fellowship with her. And so this is a study where they looked at, they analyzed electronic health record data from 73 million patients at almost 400 US hospitals. 10% of the sample had substance use disorder and 12,000 had COVID. And what they reported right now in molecular psychiatry this year is that a diagnosis of recent, so within the past year, of substance use disorder makes a person almost 9 times more likely to have a COVID diagnosis. And that was led by opiate use disorder, so 10 times more likely if you have a recent past year's diagnosis of opiate use disorder, followed by tobacco, alcohol, cocaine, and cannabis. But even a lifetime diagnosis of substance use already increases the likelihood of a COVID diagnosis, again, led by opiate use disorder, then cocaine, alcohol, and tobacco. For the people who have the COVID diagnosis, having also a substance use disorder increases morbidity and mortality. So here you can see the rates of the people with COVID. These are the death rates. So people without substance use disorder, around 7% of them died basically from these 73 million people. And then if you add the substance use disorder, it increases substantially to around 11%. And then it is explained by recent substance use disorder. Same for hospitalizations, where you have, these are all COVID patients, around 30% are hospitalized with COVID, but then it jumps robustly to around 45% hospitalizations if you also have a substance use disorder, again, explained by recent past year substance use disorder. And what you can see also is that this people with substance use disorder are clearly at higher risk of contracting and suffering worse consequences from COVID. And this is especially true for people who are African American as compared to Caucasian. And there are many different reasons that can explain something like that. So there are comorbidities and increased prevalence of asthma, chronic kidney disease, type 2 diabetes, hypertension, obesity, HIV. But there are also societal type of factors that have to do with access to healthcare, socioeconomic status, social adversity. And it is led by opioid use disorder, potentially because of the slowing of breathing that is caused by opiates, leading to hypoxemia, which is reduced blood oxygen, and that is not good for the brain, or cardiovascular or pulmonary health. And you have also, though, more infections in people with substance use disorder, the inability to keep social distancing when you have to get your drugs. And but smoking, for example, that was also leading to tobacco use disorder, that can be associated with lung damage that is caused by smoking. So clearly, it's a public health imperative when thinking about COVID to actually target people with substance use disorder as a special particular population. Okay, so what do we do in our lab? We focus on people, on recruiting people with substance use disorder, mostly cocaine, and now a new study where we look at heroin use disorder. And we put a lot of effort in those recruitment, but also then assessment of subjects in terms of psychological function, cognitive, emotional, personality function. And then we do our imaging studies. And today I'm going to talk about studies of electroencephalography and our 3T MR studies. Our subjects are, of the many studies that I'm going to show you, most of them are people who smoke crack cocaine for about 20 years, about 4 days a week. Most of them are males, and I'll get back to this point. More than half are African American. About 40 is the average age. They have high school education level and average intellectual functioning. And we always recruit healthy control subjects, and we can match them on many of these demographic variables when we compare groups, with the exception of variables that are part of the phenomenology of addiction, for example, other drug use like nicotine smoking or negative affect and depression that are always higher in addicted individuals. We can either statistically control for these variables or study their unique impact on our results. We recruit people using flyers, word of mouth, newspaper ads, but also from treatment centers, and we have wonderful collaborations across the whole entire city and outside in the whole boroughs with Samaritan Village, where Nelly Klein is on the board of directors, but also Phoenix House and other places. And I'll present results where we scan people at baseline, so about 3 weeks after detoxification, discontinuation of drug use, and then 6 months later with abstinence, and where all subjects substantially reduce their drug use. And we monitor abstinence with several ways, with monthly phone calls to the subject, to their collaterals, to the treatment center, but also at follow-ups, we have many measures to look at retrospective data reports. The presentation is organized to 4 themes because I don't have enough time to talk about all of it. And I'm going to start with the notion that cognitive function is impaired in drug-addicted individuals, and it's associated with the prefrontal cortex. So I like this study. It's actually one of the earlier studies, not from our lab, using MRI and structural MRI and voxel-based morphometry to basically, where each voxel here is assigned a probability of belonging to a certain tissue class. So it can be gray matter, white matter, or CSF. And it is based on its intensity and prior probability map. So here is a great method to quantify the actual gray matter volume in the whole brain. And here, when people across labs, across drug addictions and substance use disorders, when you compare healthy individuals to drug-addicted individuals, you see reductions in gray matter in certain brain regions. This is the ventromedial prefrontal cortex. And this is a very well replicated finding now. And you can think of it to yourself, how can it be that people with addiction have these reductions in gray matter volume in the morphological integrity of some brain regions that are so essential to our ability to function, to make the right decisions, to control our behavior, to regulate our emotions. And so let me talk about a little bit about dopamine. And all substances of abuse, all substances that are abused by humans cause this supraphysiological dopaminergic release in the brain. And so it's very high and much higher than any primary natural type of rewards like food, for example. But with chronic use, those hyperphysiological dopaminergic reactions are counteracted, are counterbalanced. And at the end result is that you have a hypodopaminergic state. So you have decreases in dopamine tonic levels in people with addiction, and which may cause persistent cellular changes resulting in reduced neuronal volume. And there is evidence for that in both human and animal studies. So in humans, it has been shown reductions in dopamine D2 receptor availability as associated with these reductions in gray matter volume. And there are other markers of neuronal cell loss that are also there in people with addiction, and reductions in brain metabolism that are associated with loss of postsynaptic dopaminergic markers. In animals, medium spiny neurons are the principal targets of dopamine terminals, and dopamine depletion in animals results in those neurons, but are with shorter and fewer spines. And I'm also going to mention that dopamine depletion, like what we think is happening in addiction, can trigger large-scale gene expression changes. But within our studies of imaging, what we actually are focusing on is behavior, human behavior. So it's good to have this background. It's what's the cellular, molecular, what are the mechanisms that can explain this type of results. But our strength is in linking those results to behavior and human behavior and cognitive function, and as it relates to the addiction itself. So here is the correlation with playing bad cards over time. So this is a task where a person has to make the right decision. They have to maximize monetary gain, and you do that by selecting an immediately smaller reward over an immediate larger one, because the immediate larger one is associated with a net loss. So people have to know that they have to sacrifice the larger reward for the smaller one if they want to win this game. But there is this correlation where the lower the gray matter in this region, in the ventromedial PFC, the lower the gray matter, the more playing bad cards over time in people with addiction who play bad. On this task, they show disadvantageous type of choice. And we think that this type of a correlation between disadvantageous choice on this cognitive simple task in the lab and its correlation with reductions in gray matter in this region may underlie the disadvantageous choices people make outside the lab, where they keep going to the same places or doing the same type of acts, using drugs despite those catastrophic consequences in their life. In our lab, Nelly Klein led this study in cocaine-addicted individuals, and she showed reductions in gray matter using the same method in a very similar region, the medial or orbitofrontal cortex, and this time showing a correlation with lifetime years of cocaine use, such that the lower the gray matter in this region here, the longer the lifetime, the chronic cocaine use. And now this region is so important not only to decision making, but also externalizing behavioral disorders, including impulsive behaviors, novelty, and together with the OFC, there were also reductions in gray matter in the dorsolateral prefrontal cortex, and together they're needed for goal-directed motivation. And I'll go back to the DLPFC later in the talk. There were also reductions, not only in the prefrontal cortex, but also the hippocampus. So for example, here you can see that the hippocampus showed correlations not only with lifetime years of cocaine use, but also lifetime years of alcohol use and lifetime years of smoking. And many of our subjects are comorbid on all of these three. And when we see this type of results, we think about projections from the OFC to the entorhinal cortex that is innervating the pyramidal cells in the hippocampus, and that together they may underlie the process that is needed for through which information about the emotional significance of stimuli is not only remembered, but also forgotten, and it is extinguished. And we think this process actually is impaired in addiction. There was a unique correlation between the OFC and lifetime years of cocaine use. And again, in animal studies, long-term stimulant use limits spine density in the orbitofrontal cortex. And we can talk about alcohol if you have questions later on, same for smoking. But the point of trying to make in these studies is that those correlations are it's very appealing for us to then assign causality. Oh, okay, so this lifetime use and chronic use of drugs leads to decreases in gray matter in these regions. But it's of course only a correlation, and there may be a third factor, at least a third one, that explains both of them. It explains both the reductions in gray matter and the propensity for long-term use of substances in a harmful way. And those can have to do with genes, and we have shown the importance of the monoamine oxidase A gene, but also that and other dopaminergic genes, and you can ask me about that after the talk. And other factors such as childhood maltreatment. Again, all of these factors that may increase a risk of a person to become addicted may be explaining both having this gray matter decreases before even substance use starts, and then propensity to use drugs again and again. So how do we go about the causality issues in our studies and being able to say, okay, maybe those factors are there in play, but is cocaine and drug use in general, does it also contribute to even increased and further loss of gray matter and decreases in function? So one way is to focus on within subject studies, and this is the retest study that I mentioned before. So here we scan people at baseline around 3 weeks after last drug use and after detoxification, and then 6 months later, when all of them have achieved a high level of abstinence and a significant reduction in drug use. And you can see how these are all cocaine addicted individuals and how the gray matter in those regions I just showed you increased with 6 months abstinence. So if you remove the immediate effect of the drug for 6 months, there is a plasticity in the brain. There is a recovery of gray matter in those regions. And again, it's still not enough to assign causality. We did not randomly assign those people to either become addicted or to then achieve abstinence. And actually most people with addiction cannot achieve abstinence for 6 months. This is a very selective sample. But if we look at animal studies, and what comes to mind when thinking about those studies are the studies by Charlie Bradbury with non-human primates, where he can assign a certain monkey group, a non-human primate group, to receive cocaine. And then he showed that chronic use of cocaine decreases gray matter in the same brain regions. And then 2 years of abstinence from cocaine is associated with increased gray matter in the same brain regions that showed reductions. So that allows us then to think about it more causally. But again, back to the human studies and our focus on behavior. And what we can do is assess at the same time that we assess gray matter in those people, we can also look at their function, at their behavior, cognitive function. And this is the same decision task that I showed you before, where you have to make advantageous decisions. You have to avoid immediate high gain. And here at baseline, people with addiction had deficits, like I showed you before, but they improve after 6 months of abstinence with no longer having any differences between the healthy control subjects. And there were a direct correlation between those increases in gray matter in the inferior frontal gyres that we need for self-control, inhibitory control. The higher the recovery in this region, the more the accuracy and the lower the percent of preservative errors on another higher order executive function task, the Wisconsin card sort test. So we think that those people with the recovery in the gray matter in these prefrontal regions and recovery in decision-making, and also correlations with this executive function that has to do with the ability to shift behavior in response to shifting reinforcement contingencies. Those are the factors that allow them then to abstain from drugs, to make the right decisions, and to shift their focus to non-drug related activities. So those studies are also making us think, when I mentioned the OFC, and the hippocampus, the DLPFC, they are making us think about, okay, are those impairments and the recovery only at the level of gray matter? What happens to white matter tracks, right? Now, we cannot use the T1 and VBM for white matter quantification. Instead, we're using DTI, the diffusion tensor imaging, which is an NMR techniques that enables the measurement of restricted diffusion of water in tissue, and it gives us the fractional, one measure is fractional anisotropy, which shows us myelination in white matter. In this study, we started not with the OFC and the hippocampus yet, but we started with the lateral abenula, because there is a lot of interest in it in the whole entire FBI community, actually, and in our lab. It's a small epithalamic structure that plays a role in both reward and aversion-related motivated behavior. And we have shown, the work, Nelly's work showed how habenula and cortical connectivity is essential for inhibitory regulation of aggressive behavior in people with intermittent explosive disorder. So here we are using basically the high myelin content in the habenula relative to the surrounding tissue in the thalamus. And we visualize it, and we use automated segmentation algorithm that was developed by Gordon Shu, who in Mount Sinai before he moved to Texas. And we basically are making it neuroanatomically correct by filtering out tracks that belong to the adjacent mediodorsal thalamus, and by removing aberrant tracks. And then we're extracting from each voxel containing at least one streamline, this fractional anisotropy measure. And all of these are then averaged across the entire tract to measure the underlying microstructure. So looking at this measure of white matter connectivity and myelin content between the lateral abenula and the PFC, we can see those are the healthy control subjects, and we can see reductions in the connectivity between those two regions in people with cocaine use disorder who are positive for cocaine in urine, so they're current users, they use cocaine in the last 3 days, and those who were abstinent, so they are not positive for cocaine, but they are addicted to cocaine with longer abstinence. And the interesting result was the correlation with age of onset, such that the lower the FA, the lower this fractional anisotropy, the more degraded the white matter tracks between those regions, the younger the age of onset of cocaine use. So maybe this degradation predispose individuals to start using drugs earlier. Okay, so back to cognitive function and behavior. And I spoke about decision making, about ability to shift, but it's really important to realize that cognitive dysfunction is as generalized in people with addiction. It's something that is not immediately apparent to us, because here we looked at tests of visual memory, verbal knowledge, verbal memory, attention, executive function. So very classical neuropsychological type of tasks. We standardized the test results to a mean of the healthy control subjects to have a mean of 0. And then this is the severity of impairment in Z-score. And when we look at cocaine addicted individuals and people with alcohol use disorder, the average severity of impairment was around less than 1 standard deviation below the healthy control mean. When you look at people with schizophrenia, their severity of impairment is much more pronounced. So although the cognitive impairment is there in people with addiction, it's not something that is as immediately observable as in people, for example, with schizophrenia, and you need those neuropsychological tools to diagnose it. But this type of minimal severity of impairment may explain the notion of addiction not being accepted as a disorder of the brain still in many, many different forums till nowadays. But it is there, it's significant, not only statistically, but also for day-to-day life. And this is a study that showed that the higher the global cognitive function, assessed by another mean, the lower the odds of attrition from treatment. So if we can somehow increase cognitive function in people with addiction, maybe we can maintain them in treatment for longer periods of time. And this is something we think about a lot in the lab of how do we increase cognitive function in people with addiction. Okay, so that was an introduction that took me quite a lot of time. So let me try to maybe speed a little bit the progress now. So in addition to this cognitive dysfunction association with PFC, we also have the IRISA model postulates that attention bias to drug-related use is enhanced, and there is a decrease in attention and response to non-drug-related cues. We can see the best way when, I'll show you, this is a result of an EEG study, and people passively view these pleasant images, horrible and pleasant images, neutral images, and drug-related images, and we quantify the late positive potential. It's an event-related potential in the ongoing EEG. So those are positive and negative deflections in the ongoing EEG that are time-locked to a certain event, which are those pictures. And when you look at healthy control subjects, we always compare pleasant images to neutral, so something reinforcing to something neutral, early and late processing times, and we can see a nice, robust response to pleasant images. This is a bottom-up automatic type of measure. We see the same thing for unpleasant images. This is a measure not of value, but of arousal, and nothing for the cocaine images. And if you look at those pictures, those type of bags will be nothing to do. Maybe it's a little sand or sugar or something like that. So it's not as arousing. But if you are a cocaine addicted individual, clearly there is an automatic arousal response to these drug-related images. And another thing that was extremely interesting in the study was the reduction in arousal response to both the pleasant images and the unpleasant images the longer a person continued looking at those pictures, as if there is a premature habituation to these emotionally evocative images in people with drug addiction. What happened 6 months later? So this is a baseline 3 weeks after detox. This is a follow-up, pleasant minus neutral, early and late response windows. You've seen that you have reductions in the LPP in response to those pleasant images. But at 6 months later, there is a recovery of processing of those pleasant images, both early but also late in looking at those images. And when we compare cocaine to pleasant images, so no longer looking at neutral images, but really contrasting drug-related images to pleasant images, which is essential in understanding addiction, we can see that at baseline, immediately after detox, there was more reaction, more arousal associated with the cocaine images as compared to the pleasant images. But with 6 months abstinence, it's flipped. And there is a reduction in that arousal. Now, this is a contrast between, it's a contrast, right? So it can be either explained by this enhances in response to the pleasant cues or reduction of responses to the cocaine cues. But you have to then listen through the whole talk to then for me to tell you which one is which, okay? And that's actually a very important point in the talk and in understanding the IRISA concept and abstinence in addiction. Okay, how else can we assess this type of drug-related reactivity? We can also use words, and we can use tasks that are performed outside of the scanner. And we modify neuropsychological tasks. So instead of asking about animals or fruits and vegetables, we ask people to call to mind and name as many drug-related words as possible in 1 minute. They can be names of drugs, people, places, or state of mind related to getting, using, or recovering drugs. So people are just telling words in 1 minute that are related to drugs. Healthy control subjects tell us over-the-counter meds. They also, while cocaine-addicted individuals tell us about paraphernalia of using drugs, and they overperform, they outperform healthy control subjects. They're the expert of this domain. And think about it in the context of those decreases in cognitive function I showed you before. So when it is related to the drug domain, you have increases in function, which is not necessarily an indication that it's something that's good for behavior. It's an attention bias measure. And the important result here was the correlation between this drug fluency that we can quantify, the drug-related fluency versus neutral fluency, tell me about animals or fruits and vegetables, in the cocaine-addicted individuals and the healthy control subjects. And we see the increase in drug-related fluency in people with addiction and not in healthy control subjects that is associated, and there was a direct correlation there, with the same response at the level of the brain, of the mesencephalon. So this is also when they're performing a word task. So they're just reading drug-related versus neutral words. And you have the same type of response in the level of the mesencephalon. So this is location of the ventral tegmental area, the substantia nigra, a major source of dopaminergic release in response to motivationally salient, conditioned cues. If you stimulate this region here, you have dopamine release in other brain regions like the striatum. And I'm reminding you that addiction is a hypodopaminergic state, but when people with addiction are exposed to drug-related cues, including words that are unique to the human, you have a strong desire for the drug, and you have increase in dopamine cell firing. And actually, it's related to cue-induced relapse to drug seeking in animal models. So maybe those words are conditioned to drug use, and people hear words all day long. Maybe they are associated then to some very automatic processes that can then lead to decrease in self-control, enhanced biases to drug-related cues and drug seeking. And right now, this is Ahmed Asseli, a postdoc in the lab, where we looked at the same region with people completely and entirely different cohort of subjects, completing an entirely different task, but again, having to do with self-control and drug-related words, and showing the same exact enhancement in the mesencephalon for drug-related words only in drug-addicted individuals and not healthy subjects. And this is in the context where the prefrontal cortex is actually reduced performing this task, and these are the cocaine-addicted individuals, the controls, and this is fMRI bold signal, and you have reductions in those regions that are so important for cognitive conflict and emotion regulation. Okay, I'm skipping self-awareness talk. We would be here a couple of hours, and straight to what do we do? So Nelly and I are clinical psychologists by training, and we are really thinking about treatment and intervention. So the first decade of our work together was focused on understanding the abnormalities vis-a-vis cognitive function and behavior in people with addiction and using those imaging results. And now we started kind of closing the loop of what do we do with that? How can we help? Can we use this mechanistic type of understanding to design interventions, or at least test the ones that may help with recovery? And this is in the context of lack of treatment of people with addiction. So this is past from 2016, people with substance use disorder do not receive treatment, 93% of them. no treatment, although they meet criteria for the disorder. If they have any mental illness, they do receive some treatment, definitely more receive treatment. With serious mental illness, most people do receive treatment. But when you combine mental health and addiction, again, you're back to 93% of people who do not receive treatment for many, many different reasons. But this is something unacceptable. So in the lab, I want to talk about a few ways that we're trying to develop things. So one, how can we reduce this attention bias to those drug-related cues? And here, while the people are looking at those images, now not just passively, we tell them either continue passively looking at them or decrease your emotional response to these cues. And we ask them to reinterpret, not just distract from these images. This is a picture of a person simulating smoking crack cocaine. And we ask them to tell themselves, for example, this is not my crack, this is a movie prop. And then we assess, after they do that, we assess their attention, and they can look at the screen wherever they want. And those images are part of the images they looked at: cocaine-related, neutral, unpleasant, and a blank for control. We also assess the ratings of wanting and craving. So gaze duration is the duration of time spent looking at one cue over the other. It assesses attention bias, and cognitive reappraisal strategies reduce gaze duration to salient stimuli in health. So first of all, we see that everybody, healthy control and people with cocaine addiction, reduce their arousal as measured by this late positive potential when they're told to decrease their emotional reactivity, which is great. So everybody is doing it and it is successful. But really the important result in this study is that only people with addiction reduce their gaze duration, the spontaneous gaze duration, after reappraising. So just reappraising those cues made them look spontaneously at the drug-related images less time than on the other images. So it gives us hope that maybe this is generalizable, and maybe this reappraisal training will then be related to their ability to reduce their spontaneous attention bias, non-trained outside the lab. We also use stimulant replacement therapy. So this is with methylphenidate. It blocks dopamine transporters, increases extracellular dopamine. It increases salience of food and math tasks in healthy controls. It bolsters cognitive functions by improving signal-to-noise ratio, optimizing activity in brain regions that support accurate task performance. Oral methylphenidate, Ritalin basically, decreases risk-taking, improving inhibitory control in other disorders that impact the prefrontal cortex, such as frontotemporal dementia, ADHD, traumatic brain injury, stroke. And we hypothesize that it will improve brain behavior function also in addiction, normalizing this hypodopaminergic state I mentioned before. And it's not that far out, because interference with binding of a drug to target, but slower pharmacokinetics, is extremely valuable in the management of heroin addiction, nicotine addiction, etc. So the same task that I showed you before, where you just have to read those drug-related words, and it was associated with reductions in the PFC in cocaine-addicted individuals. Now, in a study where we administered methylphenidate counterbalanced with placebo, double-blind design, with just one pill, 20 milligrams of this pill, one time to everybody. And you can see how methylphenidate now normalized function of the cingulate, the dorsal cingulate and the ventromedial cingulate in people with addiction as compared to the placebo condition. And this was also associated with improvement in task performance. I'm gonna skip this slide, just showing that methylphenidate is good not just for one task, it's actually for resting state, and I'll talk about it in the summary. So in addition to this cognitive reappraisal I mentioned, stimulant replacement therapy, we also are now starting with stimulation. And this is starting with transcranial direct current stimulation in cocaine addiction. So we're using this non-invasive procedure, using this device that delivers a small but continuous direct current across the scalp. So we use 2 milliamperes. It's 1/200 of what's delivered for something like ECT, electroshock convulsive therapy. And it induces polarity-dependent alterations of cortical excitability. And the key is how can we use that to then develop a self-administration type of protocol. This is so we're thinking about the remote home use. And TDCS of the dorsolateral PFC has been shown to reduce craving in cigarette smoking, marijuana, alcohol, food in healthy subjects, and cocaine. And we now have this subcontract to perform a phase 2 study using TDCS of the right DLPFC to reduce craving in drug-addicted individuals. And I'll show you results of the phase 1 study. We chose the DLPFC for many reasons. It's summarized in this Neuron paper, showing that the DLPFC as part of the executive network is enhanced during drug cue exposure as postulated by the IRISA model, but it is decreased across the board in everything else, in people with drug-addicted individuals. And I showed you also reductions in gray matter. And we showed abnormalities in this region across many of our tasks. I'm going to skip this slide. Okay, so phase 1 that we completed with the TDCS, where we recruited 29 cocaine-addicted individuals. We randomized them to sham TDCS versus real TDCS. So people are receiving this 2 milliampere electrical stimulation, excitation of the right DLPFC, and reduction of excitation in the left DLPFC. And they are receiving it for 20 minutes, 3 times a week for 5 weeks. And what we showed is a trend for reduction of drug-related craving in those people who received the real TDCS stimulation, replicating those other studies. This is a very small sample, so the fact that we have only a trend actually makes a lot of sense. We did have significant effects on increasing daytime arousal and motivation to change drug-related behavior. So to summarize, I spoke about IRISA and impaired response inhibition and Salience attribution and addiction. I spoke about cognitive impairment associations with prefrontal integrity and function and effects of chronicity of use. I spoke about attention bias to drug-related cues. It is consistent with the modified neural sensitivity to non-drug stimuli and also to monitor reward, which I did not show you, but there are similar results in alcohol use disorder. And in general, it's all consistent with the fact that the value of a drug reward is increased, but the value of non-drug reward is decreased, maybe because of adaptations to this supraphysiological stimulation by drugs. If you have compromised sensitivity to reward and relative reward, you have impairments in decision-making, impulsivity, depression, anhedonia, and impairments in insight. I did not have the chance to talk about that today. I spoke about abstinence and intervention. The reduction of drug use over 6 months period in initially treatment-seeking individuals was associated with normalization in both structures, so PFC gray matter volume, and that was associated then with better function, and also function itself, as showed by enhancement in scalp-recorded markers of salience of the pleasant of the drug stimuli. There may be also enhancement in dopaminergic function, which I did not show, but this is published. I showed that cognitive reappraisal reduces drug-related attention bias. Will it generalize to outside the lab? What will be the duration of effect? Can we enhance it with TDCS, for example, which is now what we're embarking on with our phase 2 TDCS study? I showed you that methylphenidate enhances, a stimulant, enhances the prefrontal cortex in people with cocaine addiction. I showed you only during drugs troop task, but we published and we showed it in other different tasks, inhibitory control tasks and resting state connectivity. Can we use it as a cognitive enhancer, not just over the counter administration, but really to use it to then retrain the brain in people with addiction. The TDCS of the DLPFC shows promising results. We have to replicate and validate them. Beyond highlighting brain plasticity, even after chronic use, and beyond providing hope to drug-addicted individuals, we think that those type of neuroimaging biomarkers can be useful to tracking and improving follow-up outcomes in addiction. And the length of follow-up period and the function assessed are crucial. And that's when, if you recall, I told you, remember that contrast, pleasant to cocaine, which is what's happening really in the brain. So these are animals from, these are animal studies showing the concept of incubation of craving. As a function of abstinence, you have increase in drug seeking in animals pre-trained with drugs. So if you think about it, you expect, okay, if an animal doesn't find a drug, their craving or drug seeking will decrease. But it's the opposite. It actually increases before it decreases around 6 months later. And that has been shown with cocaine addiction, heroin, alcohol. We published the same results using those late positive potentials. And as ERP skull-based measure of arousal in human cocaine-addicted individuals, this increase in cocaine cue reactivity while watching those cocaine pictures as compared to the neutral pictures, 1 month and 6 months into abstinence. This is a cross-sectional study. And the amazing thing here is that if you ask people about their drug cue reactivity, if you ask them about their craving, it decreases over time. So a person who is at 1 month or 6 months of abstinence, they feel in control. They don't have craving that they can perceive. This is also the time that they're released from treatment centers. But this is the most actually vulnerable time because their cue reactivity is so much increased. And maybe that puts them at a risk for relapse. So maybe if we can use our cognitive reappraisal or the methylphenidate or the TDCS, but at the right time, this is when we have to actually target this cue reactivity, reducing it. And the question then, can we enhance then abstinence and lengthen it and can then give people more control over their behavior? So, but here what it means is that drug-related cue reactivity is increasing, but we were able to also enhance non-drug alternative response that also may counterbalance this type of cue reactivity results. And we now have results of longitudinal studies. So we scan people 5 times in a year doing these ERP studies longitudinally, and we have the same basically pattern of cue-induced incubation of craving. The point of this slide is just that every person has a healthy state. Even people with severe addiction will have a healthy state where the drug-related functions are contained within the non-drug related functions, and they have the ability to stop, that they lose when they crave in withdrawal, and especially then going into intoxication and binging. So how can we get a person back again and again to their healthy state, in addition to cognitive reappraisal, methylphenidate, TDCS? And here, this is another study that we're starting now as we speak, where we are going to deploy mindfulness group therapy that includes savoring of non-drug related rewards and reappraisal in an 8-week type of group therapy of people with opiate addiction. And we're going to do imaging before and after this group therapy and longitudinal follow-up. And we're starting it at the Samaritan Village facility because they allowed us the space we need, especially now, where we need social distancing to perform not only the recruitment, the screening, but the actual intervention in a safe way. And another point I would like to make is that all of it that I told you is true for men. Most of our studies include men, and not only ours. We don't know about gender differences. So these are we reviewed all of these fMRI studies in addiction, and most studies did not include any women or had very low number of women. Only 7 out of 105 studies were powered enough to perform gender comparisons in people with addiction, focusing on fMRI in cognitive function. So we really don't know much about cognitive and emotional function and the substrates that differentiate the sexes. We know that less women become addicted, but those who do show much more severe patterns of addiction. And maybe the insulin involved, I'm going to skip that because I want to give some time for questions. And I want to thank my amazing, wonderful partner, Nelly Alia Klein, partner in crime, and the amazing group we have who are working extremely hard despite all of this stress that we're currently under. So thank you for your attention.
Thank you very much, Rita. That was a wonderful overview of your research program. I enjoyed that a lot. And thanks for leaving time for questions. So let me just, if you could unshare your screen, that way we could look at each other. And we have time for questions, and I would encourage anyone who's interested to unmute themselves and speak up. Don't be bashful. Any questions? So Rita, I'll ask the first question. I'm really glad you highlighted the study with Mohammed and you on incubation toward the end of your talk, because that's been a question that I've had. So much of the rodent literature talks about incubation, but it's been in general difficult to demonstrate that in humans. So I think your study may be one of the first or best to establish that. So I was wondering if you could talk more about the whole notion of incubation and what kinds of behavioral features you think do show sensitization over time before they then recede after longer withdrawal.
Yeah, so thanks for the question. So basically, yes, our study was the first to document this incubation concept using objective measures. So there were other studies by Harriet DeWitt, for example, where she used self-reported craving, cue-induced craving, and she showed the similar results. But in ours, it was the first time that we used an objective marker of brain function. And this was cross-sectional. So of course, it's just taking all of the data we had and then dividing it by those bins that we followed from the animal studies, and then seeing this result that was very remarkable, especially vis-a-vis the self-reports that showed this clear decline. And now that we performed this study longitudinally, so we have for every subject coming back every 3 months. And again, we see the same incubation process. But in the human world, it's really actually, and Mohammed can step in, it's so because we're analyzing this data as we speak, it's really not trivial, because we can only determine abstinence at the first time they come in, right? This is a naturalistic study of people who are either inpatients or outpatients. They're mostly treatment seeking, or they're abstinent, they're trying to maintain abstinence. But then naturalistically, many of them are using drugs. So how do you, in animal studies, you can say, okay, this is the last time the animal is receiving any drugs whatsoever. And then you can track the withdrawal effect very cleanly. For us, it is much more complicated to do that. And we statistically control for it. We look at it in different ways. We have to really still explore it. But I'm not sure if it is a sensitization type of phenomena that actually there was no documentation to it in human research at all, that there is any sensitization to cocaine cues in humans. I think those are different concepts, the sensitization versus cue reactivity, but still, it's something that definitely we need to explore.
That's great. I see Daniela raising her electronic hand.
Yes, thanks, Rita. Really interesting. So you talked a lot about decision making, and I was wondering how could this relate to their memories? I was wondering if they have memory impairments, because just anecdotally, or my impression is that the addicted population usually have less access or even less memories during their substance use, and sometimes they don't even access childhood memories. And this could affect how they make decisions in the present and predict the future. So how would you incorporate memory into decision making? Is there any evidence regarding that issue?
Incorporating memory into decision making, that is a very interesting concept. Yeah.
Or even is there any data on how, if there are any memory impairments?
On memory. There is definitely on memory and learning in drug addicted individuals. We have the study of extinction, so the ability to actually extinguish conditioned associations, and that has to do with forming memories. And there is clear decrement in the ability of the VMPFC to extinguish those drug-related cues. So although you have detriments in memory functions to non-drug-related cues, if anything, you have a resistance to extinction of drug-related cues because of the impact of on the hippocampus, for example, in terms of gray matter, but also function, especially with alcohol being a clear neurotoxic. But in terms of recalling childhood memories, The problem with, and that's, it goes back actually to also the issue of quantifying drug use in humans. Again, going back to also Eric's point, when a person starts binging, clearly it affects their memory. Any alcohol binge, anybody who did any alcohol binging in their youth knows the effect. You cannot recall how many drinks you had after a certain amount. So there is clearly an inability to form new learning and memory in a binging episode. But does that affect then remote memories? It has to be pretty severe degradation. And I think in alcohol use disorder, you can see that. So it differs by also the drug of abuse. In cocaine addicted individuals, we don't see it as much, and we can control for it statistically. But designing a task where you can look at both simultaneously, that will be a very interesting way to go. Very naturalistic, or more naturalistic than our more simple task.
Thanks.
Yeah, hi, Rita. This is Anli. Thank you very much for the great talk. Just to follow up Eric's question about the incubation, do you observe a lot of polysubstance use among this population, and what happened to the use to other substance during that period?
Yeah, so here we selected people, they met criteria for cocaine dependence and abuse. And most of them are also nicotine smokers, and many of them meet criteria for alcohol use disorder. There is nothing else, and the results were clearly influenced by the cocaine use disorder and not anything else. We do check for that.
Yeah.
But we did not, the way to go about it would be to recruit people who meet criteria for several substances, let's say.
Right.
And to really do it more in a standardized way. So our focus was on stimulant use. Now we're starting this mindfulness study in opiate use disorder, and we're starting the sex differences study in people who are cocaine use disorder or with opiate use disorder.
Right.
So there we will be able to compare the effect of drugs, of different drugs, because clearly cocaine and opiates, for example, have some very clear differences on the medium spinal neurons, for example, in the OFC itself,
Right.
and projections to the OFC. So it's something to explore still.
Yeah, because in my study with this opioid use disorder, during the antagonist treatment period, they don't use any opioid because they cannot get high from opioid. And we see the cocaine use actually shoot up. So, I mean, because, these patients are, most of them, I think over 70% of them polysubstance use.
Yeah, so we don't have polysubstance users.
So that's
okay. Not that many. We don't. We actually we do not include them.
Yeah. And the smoking, it doesn't affect their smoking.
Smoking is something we always look at, but it's it's clearly something that differs always between the cocaine users and healthy control subjects. Right. We can look though at the amount of smoking. Sometimes it differs, sometimes it does not. We can statistically control for it, but we always it's a real limitation. And we need to recruit more healthy control subjects who smoke nicotine, clearly.
Yeah.
But based on all of our analysis and the statistical controls and the correlations, etc., those results, in my mind, are driven more by the cocaine use disorder. Mm-hmm. But again, those are things that in a nutshell, it's a field that we still are so much in its infancy. Right?
I know. There's so many variables there too.
And it's it's so much more complicated than in animal models where their history of drug use. Right. Only one drug, nothing else.
Exactly. And in animal studies, any time an investigator proposes to study more than one drug at a time is criticized by the review committee for studies that are too complicated, even though the human syndrome is almost always polydrug use.
Although polydrug use is different than polydrug substance use disorder. Yeah. So people who are using many drugs, they still meet criteria for cocaine and nothing else, let's say nicotine. But polysubstance use is when they're indistinguishably using whatever they can have. And that's a little different. And that would be also interesting, but that's a different animal, I think.
Yeah. Well, listen, we're at the end of the hour. Rita, thank you again for a great talk, another great cyber series lecture. And thank you all for joining us. Have a great rest of the week and weekend. Bye-bye.